Morning. How's everybody doing?
Good.
How are you guys coping? 5 weeks in.
Coping with just things in general?
Yeah, just things in general.
Not too bad. Trying to keep everything as normal as possible.
I mean, school in particular, but life in general.
School's been interesting. It's just
how so?
This is weird to get used to everything online because some of my classes were like meet in person, like we're scheduled to, and then on the second will get changed, and then our homework gets changed, like the format and everything. So it's just challenging to keep up with everything a little bit.
Yeah, it is, isn't it? I'm experiencing that with my biology class because we're having to revamp lab because normally what we would do in lab, we would spend the entire semester doing, and we can't do that because students are going to be gone after Thanksgiving, so we lose that time after Thanksgiving. So we've been struggling to kind of preserve as much of the lab experience as we can, while also making it doable in the time that we have available to us, which is proving to be more challenging than we thought. So hopefully after today, my workload in kind of dealing with lab in my Bio 133 class is going to go down because we will have kind of gotten over this revamping hump and things will begin to settle down. I've been feeling a little frazzled lately, and I was just wondering if you guys were feeling the same level of frazzledness and discontent with the way life is right now. So anyway, how are you guys doing? Other than Winston, who already chimed in. I think that was Winston, right?
Yeah, that was me.
Okay.
Things are going fine. I just wish this COVID stuff would just end, honestly.
Yeah, it doesn't. Well, most people I think are in that camp. One thing I have been shocked at is the ability of William Jewell College to bring students back to campus and not just all of a sudden become a little cluster of COVID. I was one of the skeptics on campus of doing this, and I am pleasantly surprised to say I was just wrong. So yeah, it's nice to admit that you're wrong and have that be an unmitigated good thing. How about the rest of you?
It's pretty good. It's just like everybody else has already said, it's just so weird, and it's like hard to keep track like which class is in person today and which one's online and which one's but
so have you guys, when you accepted the Zoom meeting, did you download it to your calendar? Yeah, I'm finding the Outlook calendar to be my savior during this time because everything is on it. And so if you haven't done that, it's a good idea. And you can actually add things to your calendar that aren't Zoom meetings. And so I had never really used Outlook's calendar before this, but the nice thing about it, let me let me just do this because I'm about to have to share my screen with you guys anyway. Is that it's just kind of keeping me straight a little bit. So when I look at my calendar, this is what my calendar looks like, and it's like, oh, I've got CTI 2:59. That's a Zoom call, so a Zoom meeting. So, and then I can just click on that, and there's the link. And so, you know, any little thing you can do to kind of keep yourself sane would be a good thing. And Zoom hates me. It's gotten rid of everybody now. Nobody's on my screen. Oh, there you all are. Okay, cool. Awesome. All right. I put some things on the Moodle site for today. I don't know that we're going to get to the Jared Diamond chapter, but I put a chapter from a guy named Jared Diamond from a book called Guns, Germs, and Steel, which was written in 1997. It was controversial at the time. It remains controversial, I think, to this day. Jared Diamond is an evolutionary biologist. He's a very good evolutionary biologist. He was at UCLA for many years and was in the Department of Ecology and Evolutionary Biology, and has since then shifted over to the Department of Geography, actually. And he's a MacArthur genius. He has written a ton of really good papers in the field of evolutionary biology and biogeography. And he has always had very wide-ranging interests. And one of his interests is the history of humans. And so in this book, he takes an ecological and evolutionary perspective to the rise and fall of cultures. And so in that way, it was provocative because he was using the tools of evolutionary biology to study areas outside of evolutionary biology, mainly the history, anthropology, archaeology. And as a result of his evolutionary and ecological perspective, he comes up with some different explanations for kind of why the world turned out the way it is from a human perspective. And so there are some interesting ideas in that book. And they clue in, they kind of tie in with the things that we're talking about right now, which is the evolutionary origin and the early distribution of these crop species that we have come to rely on. So, you know, the question could be asked, why do we have a food economy that is based on wheat and corn and soybeans and things like this, and beef cattle and chickens, rather than other types of grain crops or other plant crops, and, you know, not zebras and rhinoceri? And so Jared Diamond has some explanations for this that extend beyond just kind of economics and cultures, but actually gets back to the natural communities of organisms, plants and animals, that were in places on the face of the earth where humans actually also co-occurred, and why in some cases those things led to the rise of city-states and nations, and in other places they didn't. So anyway, it's an interesting chapter. So if you haven't read it yet, definitely read it, because we will get to it at least by Friday, even if we don't get to it today. So are you still seeing my screen? You are seeing my screen, my very cluttered screen. Do you have questions over what we were talking about last time? I was attempting to introduce you to the idea of phylogenetic systematics and how biologists can recreate or reconstruct the evolutionary history of a group of organisms. Any questions about that? All right, then I'll proceed as if you guys are understanding. So one of the things I didn't talk about, and I should talk about this every time I show a tree like this, is that you can think of this figure as having a y-axis and an x-axis. And the x-axis is time. And so the time goes from the past at the far left of the axis and the present at the far right of the axis. And the y-axis is divergence. In this case, it would be phenotypic divergence. So whatever phenotype you might be thinking of, whether it's a physiological phenotype, whether it's a morphological phenotype, what something looks like, the y-axis is how different are these organisms. The x-axis is how has that divergence developed over time from the past to the present. So another way of thinking about this is to think about this as there was a species somewhere back in time. At some point, it broke into two different groups, two different populations. Those populations evolved over time. In one case, the lineage up above just kind of keeps evolving down its own evolutionary trajectory, but this species then divides into a couple of different populations and gives rise to new species C. So each of these branches represents an ancestral species to all the descendant species that come after. Species C branches into a couple of new species, D and E branch into a couple of new species, and over time, we finally end up with the species that we see now, species I, J, K, L, M, N, O, P, and Q. But all of these other species that are represented by A, B, C, D, E, F, G, H, these are all ancestral species. And in some cases, we might find these species in the fossil record, but in most cases, we infer what these species would have been like by looking at the extant species that are the descendants of that species. So we create the topology of the tree, we create the shape of the tree from things like the character matrix that we looked at last time, but then we can get insight as to what these ancestral species were like by looking at the distribution of characters in the descendant species, because presumably they got many of these characters from the ancestral species. And so time is on the x-axis and morphological or physiological divergence is on the y-axis. So I showed you this map that basically just shows you the biogeographic origins of a bunch of different kinds of domestic plants and animals. Domestic plants and animals that have been incorporated into our food supply. And a lot of what we're going to be doing today is exploring that in more detail, and also doing that on Friday to some degree. And I introduced you last time to the cattle. And so one of the groups that you have to pay attention to if you're going to pay attention to food in the world is humans' use of cattle, both as beasts of burden for plowing fields and things like this, as well as a source for milk and meat. And so I mentioned to you the two types of buffalo, an African buffalo and a water buffalo from Southeast Asia. I showed you the bison and the wisent, which are northern landmass organisms, the bison in North America and the wisent in what we call the Palearctic, which is Northern Europe and Russia. The yak, which is found in the Himalayas, the banteng, which is found in Australia, the gar, which is found in Southeast Asia, and then we have modern beef cattle, which are found in the Palearctic or found in the subcontinent of India. Well, when we think about this, the earliest, so the outgroup that we would use for reconstructing a phylogeny of cattle is this thing called the bongo, which is an animal in Africa. And then the next oldest groups to have evolved are the water buffalo. And so we have two branches of water buffalo, a Southeast Asian water buffalo and an African water buffalo. And then the companion species, the companion group to that ultimately gave rise to yaks and the bison. And so one of the things about yaks and bison is they're all kind of northern oriented species. The Himalayas is on the north side of India, south part of Russia. and bison are all across the northern part of North America and the Old World, Northern Europe, and Russia. And then we have the banteng, which is in Australia, and then the gar, which is in Southeast Asia, and then we have the auroch, which is Bos primigenius. This is the ancient predecessor of modern cattle, which are the zebu, which is in India, Bos indicus, and then Bos taurus, which is Fertile Crescent and European. And so this is the most up-to-date phylogeny of these groups of hoof stock that people have used for agriculture. And I mentioned earlier that in some cases we have fossil evidence for the wild progenitors of modern organisms. And so we have actually quite a lot of fossil evidence for Bos primigenius. And so this particular ancestor we know quite a lot about because we have found its bones in caves and things like this of early humans. And so the development of modern cattle is a relatively recent sort of development. These other organisms have diverged mainly due to natural selection, not due to artificial selection. But as you'll see here in a moment, the modern cattle have actually undergone quite a lot of artificial selection over time. So these are the names. The next slide actually superimposes the pictures of each of these species onto this phylogenetic tree, and I think this will be most useful to you in seeing that the tree is actually probably accurate. So when we put these up against one another, so this is the tree, and you can see that there's a lot of morphological resemblance between the two species of water buffaloes. There's quite a lot of morphological similarity between the two species of bison, the bison and the wisent. There's the yak. It looks different from these guys, but one of the things that you'll notice about it is that it's very thick in the front, very less heavy in the back, just as the bison are. It has a boss, a shoulder hump, just like the bison do as well. And so once again, there are certain resemblances, even though that breaking off of the yak lineage occurs early in the evolution of this particular lineage of organisms. The banteng is just kind of an interesting looking animal with short front legs and long hind legs from Australia. Then we have the gar, which is this very beefy buffalo-looking creature from Southeast Asia. And then we have the domestic cattle, and then we have an artist's rendition of what Bos primigenius looks like. Now you'll notice that the zebu, Bos indicus, has this boss also, this shoulder hump on its back. It's much more pronounced in terms of how radically compressed it is. It's very short but taller than the shoulder hump of the yaks and the bison and the wisent. And so these are what they look like, but the topology of this tree was actually generated by genetic data. And so people went out and sampled tissues from all of these different types of organisms, and rather than using morphological evidence to create this phylogenetic tree, they actually used genetic data. And so it's nice when you have a genetic tree that then when you look at the morphology of the organisms, you could see, okay, well, that tree actually does do a pretty good job of describing the evolutionary history of these organisms. Of all of these organisms, the one that has undergone the most change is the domestic cow, Bos taurus or Bos indicus. Depending on who you talk to, people will put those two things in the same species, and in some cases, they split them out into two different species. I've kind of superimposed this tree on the different kinds of organisms that are here. And so here at the very beginning of the tree, you have the bongo, which is an African species, and then you have I have to go back to the tree. Oh, you have the bongo, and then you have the bongo as the outgroup, and then you have the African buffalo and the Southeast Asian buffalo. Then you have this group here, which is the two varieties of cattle and the gar and the bantang. And then here you have the North American bison, the wisent, and in the Himalayas, the yak. And so this is kind of just the evolutionary tree superimposed on the geography. Sometimes people find this useful, sometimes people just find it confusing. If you find it confusing, don't worry about it. If you find it helpful, then pay attention to it. So Bos primigenius, which is this ancestral species to our modern cattle, had a wide-ranging geographic range, and as climate changed, this population became disjunct, such that there was a European version of it and then an Indian subcontinent version of it. And so the zebu, which is this animal here, is the Indian subcontinent version of things. And anytime you see an organism, a cattle with a little bit of a shoulder boss here, that's going to be a species that either descends directly from zebus and has been modified, or has been crossbred with Bos taurus. And so actually, in this case, all of these breeds are breeds that have been crossed with Bos taurus. This is a Hereford, a typical Bos taurus sort of animal, and there are varieties of this. Once again, this is a version of Bos taurus that has been crossed with the zebu. This is one of my favorite domesticated animals. Do you guys know what that is in the upper right-hand corner? Any college football fans out there?
Longhorn.
Yeah, so this is a Texas Longhorn. I'm from Texas originally. I grew up in places where you could see these things roaming freely. And so what happened was the Spanish brought domesticated cattle over to the New World when they colonized the New World. And after they left, after they were kind of displaced by other groups, i.e. Americans, European Anglo-Americans coming from the United States, they left behind a lot of cattle, and these cattle went feral, and they underwent natural selection, and the natural selection transformed them into a very different looking breed of cattle. And so this is a Longhorn. It's characterized by long horns mainly. They also have really big long heads. They're relatively lean. They don't pack on a lot of fat. And one of the advantages of them for cattle ranching is they're very well adapted to living on very poor forage and living in drought-prone areas. And so Longhorns have made a little bit of a resurgence in the last couple of decades because of they possess certain characteristics. One of the characteristics being able to maintain their weight while eating really crummy food and not getting very much to drink. This is actually a world champion Longhorn that is raised by a faculty member at the University of Texas at Austin, a guy named David Hillis, who is also a MacArthur Fellow. And one of the things that he did with his MacArthur Fellowship was he went about the process of looking into the evolutionary biology of Longhorns and looking at their genetics and looking at how you can essentially improve the breed. And after a relatively short period of time of managing a herd of Longhorns, he produced some truly remarkable specimens. This is one of the ones that has been among the best. So domesticated animals can undergo artificial selection, but domesticated animals can also undergo natural selection when they are, left to run feral. This lower right animal here, this is actually a Spanish fighting bull. So this is a bull that has been bred not for beef, not for milk or anything else, but is bred for being used in bullfights. And you can see that its structure is somewhat different than animals that is built for carrying weight on the hoof. They're leaner. They're kind of squarer in the back. They're much heavier up front than some of these other animals are, and all of that is characteristics that are used that are designed for creating good fighting bulls. Once again, a couple of other Spanish fighting bulls. There are other branches of the Bos taurus tree that have been developed for milk production. The things that you will notice about these guys is that they have very large udders. They've been selected for milk production. They don't carry a lot of weight on the bone, and so they're not very good for growing for beef production like a Hereford would be, or a Longhorn even for that matter. And so they're leaner in body, but they have invested way more in the development of the udder, which makes sense because that's what a milk farmer would want. This is a Jersey, and this is, man, I forget that breed at the moment. So these are just examples of cattle. This is the end point of this evolutionary branching of major bovine species that have been co-opted by humans. So this is Sus scrofa. This is the geographic range of Sus scrofa, which is the boar, the wild boar, which was developed into the pig. So they were found native throughout Northern Europe, and then also all throughout Southeast Asia. They've become introduced into the United States, and they have become introduced into New Guinea and Australia. This is what Sus scrofa looks like in its natural state. This is a wild boar. They tend to have really coarse hair. They have this hump over the shoulders once again. They tend to have long tusks. We have taken wild boar, though, and through domestication, we have transformed it into a wide variety of breeds of pig. Berkshire, which is a Northern European breed, you can see scale, the pig relative to this human. That human is standing back a little way, so the human is smaller than it would look if it was standing right next to it. But we have bred some pig breeds to be quite large. Vietnamese potbelly pig has become a popular pet among some people in the United States. Most of the pork that you would be eating here in the United States is from the American landrace, this individual here. If you were to go to a modern hog farm, you would almost certainly find this particular breed being the one that is used for pork production in the United States. So Sus scrofa is the wild progenitor of pigs. And they have a really interesting evolutionary history, once again, based on their genetics. And there are two lineages. One lineage went into the Palearctic, Russia and Northern Europe. And there was another lineage in Southeast Asia. And in both of these lineages, there was domestication. And there was one line of domestication that occurred in the Fertile Crescent. And then those organisms actually moved out of the Fertile Crescent into Europe. At the same time, the Southeast Asian species was domesticated in Southeast Asia and stayed in Southeast Asia. And this Palearctic branch of the evolutionary tree just stayed separate until later on, there was a secondary domestication where people brought the South Asian variety of pigs from the Fertile Crescent into Europe. But when they got to Europe, as humans expanded into Europe, they had their own pigs that they were bringing with them, but they evidently found that the pigs that were already there served as better stock, probably because they had undergone local adaptation to the environment of Northern Europe. And so what happened with modern pigs is they basically discontinued using the Fertile Crescent. What happened there? Get off my screen. They got rid of the Fertile Crescent stock and basically secondarily domesticated pigs that were already there in Northern Europe, which leads to this really complicated evolutionary tree for pigs. So this line essentially ultimately died out and is no longer found because all of the pigs that we have now that we use in Europe and for the most part North America are due to the secondary domestication once these human beings moved out of the Fertile Crescent into Europe and found pigs that were already kind of pre-adapted for that environment. So we now have kind of two major evolutionary branches, this Palearctic branch and this Asian swine branch that is found throughout China, Southeast Asia, Malaysia, Indonesia. So essentially a widespread species, two lineages though, a Southeast Asian lineage and a Fertile Crescent lineage. This Fertile Crescent lineage moved out of Europe, out of the Fertile Crescent into Europe, but then was abandoned when these people who migrated from the Fertile Crescent into Europe found pigs that were already there, that were better suited to that environment. Therefore, that Fertile Crescent lineage died out and was replaced by this European lineage. And then, of course, humans have moved pigs around because pigs are good for food. And so that's what those other introductions in Australia, Papua New Guinea, and the United States have been about. The chicken, Gallus gallus. If you are an ornithologist and you're studying chickens in the wild, you would call this the jungle fowl. And this is actually a picture of a jungle fowl in its natural habitat. In 2010, I was in Indonesia, and as I was hiking around the island of Komodo, I was surprised to see this island where there basically aren't any people. There's one little section of the island where there's the national park headquarters, but the rest of the island is uninhabited. And yet we ran across a jungle fowl out in the dry forests of Indonesia. And these weren't escapees from people's farms. These are actually just chickens living their lives out in nature. And so their range ultimately is Southeast Asia and into Indonesia, the Malay Archipelago. And of course, they've been introduced all over the world as a food animal, which is not reflected in this particular map. Over time, they have undergone radical changes. In some cases, they are grown for meat, like a leghorn. In many cases, they are raised for egg production, which is the case of a Rhode Island Red and light Brahma. I've altered this PowerPoint from the one before in a couple of ways, one of which is I got rid of whatever chicken was there before, and I put light Brahmas because now I have a flock of light Brahmas at my house in my backyard in a very nice chicken coop and run. And so some varieties have been bred for meat production, some varieties have been bred for egg production, and some varieties have been bred simply because humans are crazy and they think it's cool for chickens to have really long, elaborate tails. This is particularly a thing in Asia, Japan, and China, where they have bred these show chickens that have these really elaborate long feathers, long tail feathers. And so in some cases, we've bred domesticated animals for food production, and in other cases, we've just bred them for show. But this represents kind of the power of artificial selection at changing the phenotype of a domesticated animal. You can tell that that's still a chicken. You can tell that all of these chickens are probably related to this wild progenitor, but the ability of humans to alter organisms through just simple artificial selection is quite powerful. Also, sheep and goats. And so Ovis is the genus of the sheep. And so Ovis musimon and Ovis orientalis are these two species. Ovis musimon is a European version of the sheep, and Ovis orientalis is a more Asian version of sheep. The European mouflon and the Asian mouflon You might be familiar with bighorn sheep here in the United States. These are the old world equivalents of those. And so these have been domesticated. So this is the wild progenitor. These are Oxford sheep that are in England. These are merino sheep that are grown primarily for their high quality wool. And then these, this is desert painted sheep. And this obviously is a sheep that is adapted to hot, arid environments. And so once again, the wild progenitor and the various, some examples of the various breeds that have come out of this. You can still find wild mouflon out in the wild. And so this is one of the situations where the wild progenitor still exists, just like the chicken and the the chicken and the pigs. The wild progenitor of modern beef cattle obviously is extinct. So once again, we have a phylogenetic tree that has been created. It's not super informative, except that there have been, once again, two different domestications of these species, such that you have Ovis orientalis and Ovis aries that are closely related to one another, and then you have Ovis musimon and another branch of Ovis aries that are closely related to one another. So these two species that we commonly use for for sheep raising are distinctive, but there's been a lot of interbreeding with this other variety of sheep. And once again, that's not common. We're going to see this again when we talk about wheat. People modifying species by breeding domesticated varieties with wild varieties to get more variation. Capra is the genus of the goats. Capus aegagrus rather. Once again, found throughout the Fertile Crescent region, into Europe, and into the subcontinent of India. Capra hircus is the one that we have primarily domesticated, and these are just the wild progenitors that are closely related to it. And so those are, I think, all of the groups. Oh, okay. Hold on. I didn't realize I had combined these. I guess I did combine these. All right. So as you flip through these maps, this map here, and this map here, this map, this map, and this map, what do you notice about all of these maps? What similarities and differences are there?
They're all in the same area.
Yeah, there are a lot of organisms that are all found in this particular area. And so Europe, the Indian subcontinent, and the Fertile Crescent region, what was traditionally known as the Fertile Crescent, which is also sometimes referred to as the dawn of agriculture, the birthplace of agriculture. And so that will become important here in a little while, because the distribution of organisms actually turns out to be very important for the development of human societies. And the fact that a lot of these animals that ended up being domesticated actually has importance for Jared Diamond's ideas on the development of human culture. I don't know why it was doing that. So now what I want to do is talk to you about the plant side of things. So there's another PowerPoint in today's materials on Moodle, and this PowerPoint deals with the evolutionary history of domesticated plant crops. So we've talked about animals, the major animal groups. Now we need to talk about the major plant groups. So the goal of this is very similar to the goals of the other one, which is to understand the relationship between the biogeographic distribution of crops and the crop mixture that we currently rely on in agriculture. Important to understand how wheat and corn developed into the crops they are today from their wild progenitors. We're also going to talk briefly about soybeans, but soybeans have been less studied than wheat and corn have been, so there's less to talk about there. And then we'll talk about Jared Diamond's views on the importance of plant and animal distributions in the development of human cultures. So this is a figure from your book, and it's showing you the early centers of agriculture. So we have the Fertile Crescent, which you probably read about in a history class or a social studies class or something like this in public school. At least I hope you did. We have a rise of agriculture that was largely based around rice and pigs in China. We have Mesoamerica, which was an agricultural center based on corn, squash, and beans. We have the highlands of South America, the Andean areas, area that was based largely on the development of potatoes and llamas, but they also have other food sources that they relied upon, llamas and vicuñas. And then we have the New Guinean highlands, which was an agricultural area in a plateau that is at the top of the island of New Guinea. And this was the natural place where sugarcane evolved. If you look in Jared Diamond's chapter, you'll see a very similar map, but it has some additional centers of agriculture. So we have the eastern United States, we have Mesoamerica, we have the Andes, but then he adds an Amazonian region of agriculture, which is relatively recently put forth as an area of agriculture based on some recent archaeological evidence. We have the highlands of New Guinea, we have China, we have the Fertile Crescent, but then he also adds the Sahel and West Africa and Ethiopia as agricultural centers. And he puts question marks here because right now the Sahel is a prominent agricultural center in sub-Saharan Africa. It's right here at the border of the Saharan desert, so it's kind of a mesic area. It's not too wet, but also not too dry. There's a band of tropical rainforest that is just south of that, and there's a desert that is just north of that. So it's in this narrow band of not too dry, not too wet. They grow a lot of wheat there traditionally. And so one of the question marks here is because one of the questions is, where did they get wheat to establish this agricultural center? Well, they probably got that wheat from the Fertile Crescent. So through trade. Similar situation with cattle in Ethiopia. Ethiopian tribes are well known for their cattle raising abilities, but once again, they probably did not get these cattle because cattle were indigenous to Africa. They probably got these cattle through overland trade. So these are kind of secondary centers of agriculture rather than primary. Fertile Crescent agriculture arose because wheat was there, cattle were there, sheep were there, pigs were there, everything was there that you needed to get agriculture started. The Ethiopian center and the Sahel center, not so much. Let's see, bananas also are from New Guinea originally. So bananas and sugarcane. So his map is a little more complete because it includes not only primary centers of agricultural development, but also these secondary centers. So we can look at where different plant crops came from. Among the ones that we're going to talk about a lot in this class is corn, which comes from, once again, Mesoamerica. We'll talk about rice, some that came from Southeast Asia. Soybeans come from Southeast Asia. And then wheat, rye, sorghum, barley, and wheat all come from this area around the Fertile Crescent. And so the distribution of potential food sources is non-random in terms of where in the world these things came from. Lesser crops that we have, things like tobacco and potatoes. Potatoes, once again, I've mentioned, come from the Andes. Tobacco comes from the Andes. Sugar beets come from Europe. But a lot of these things don't make up large portions of our food supply. Corn, soybeans, and wheat make up large chunks of our food supply. And of course, for us in America, sugarcane makes up a large source of food supply, but had a very limited distribution naturally. So the major crops that we're going to talk about in this class repeatedly over and over again, and you probably have already sensed this from reading the Omnivore's Dilemma and then also hearing me talk, is corn, soybeans, and wheat. These are three major crops in the United States. Sugarcane used to be more of a major crop, but now most of the sugar that we get is from sugar beets rather than sugarcane. For whatever reason, it's a much less labor-intensive plant to extract sugar from. So most of the sugar, table sugar that you get is now from sugar beets rather than sugarcane. So corn. This is an evolutionary model for corn, an evolutionary tree for corn from proceeding in the National Academy of Sciences article about 10 years old now. But basically, this shows you a lot of the different grain species that have been important in the development of agriculture. So we have sorghum, we have corn, we have rice, we have wheat. And so you can see here, this particular article is primarily around, primarily centered on corn. But what you see is that corn occurred here, and there's a thing called retroposon invasion. Well, it turns out that was really important for the development and evolution of corn as a crop. And so this is something that very possibly happened in Teosinte that then led to the development of corn, which is ZMAs. We'll talk about wheat later, but once again, it's also in this evolutionary history of grain crops. But right now we're going to concentrate on corn. And one of the points of this particular paper is that we've already talked a lot about this transition from Teosinte to corn and how it's undergone this radical transformation in terms of phenotype. The size of the ear has increased, the diameter of the ear has increased, the number of kernels on the ear has increased. But most of this has been due to gene duplication in corn. And so this idea of what a retrotransposon is, let me just explain what this is. So this diagram here basically kind of graphs out what's happening. Remember that when DNA is being used to ultimately make a protein, you have to take the DNA and you have to transcribe it into RNA. And then this messenger RNA is used as a template for making an amino acid sequence, which ultimately becomes a protein. And so this is the central dogma. We go from DNA to messenger RNA through transcription. And then we take messenger RNA, use that as a template to assign the order of amino acids using the codon sequence. And this is referred to as translation. But there's another way that genetic material can be manipulated, and that's that some organisms produce a substance called reverse transcriptase, and this results in reverse transcription from messenger RNA back into DNA. So you can use DNA as a template to make RNA, but some organisms have the ability to take RNA and use it as a template for making DNA. And so reverse transcriptase is an enzyme that does reverse transcription. And so what happens in corn, and evidently this is a really common process in corn, is when this gene in the DNA of corn gets transcribed, it goes to the ribosome and gets translated into a protein. And one of the things about RNA is it generally doesn't stick around very long because it gets broken down, because it's just a temporary molecule. You want to keep it around long enough to make some protein from it, but then it gets degraded relatively quickly. But in some species, what happens is that messenger RNA, while it's out in the cytoplasm, gets reverse transcribed back into DNA. And when it gets reverse transcribed back into DNA, this ribonucleoprotein complex will actually reinsert it back into the genome of the organism that it's in. And when it does this, it's created a new insert of DNA in a separate part of the genome. So now this DNA that there was only one copy of, now there's two copies of it. And so one of the things that has happened in corn that has been very important in corn developing such radical phenotypic change is the fact that it's undergone a lot of duplication. And part of it is this type of duplication, and part of it is just actually sections of DNA breaking off and moving around the genome. And transposons in general, transposons in general were discovered by this woman named Barbara McClintock back in the 50s. And she ended up being the first woman to win the Nobel Prize, totally unassisted by a man. There were women before her who won Nobel Prizes, but they were always co-recipients of the prize with a team of men typically. She was the first one to win it solo as a woman. And it was because of her discovery of these types of methods of duplicating and moving around genes. And she discovered this in corn. And when she got to later parts of her career, she became not only interested in the genetics of corn, but she also became interested in the evolutionary history of corn. And so she actually moved to Central America, South America, and did a lot of research looking into wild varieties of corn, trying to identify what the wild progenitor of corn was. And so a lot of what we know that I talk about in this class about the early evolutionary origins of corn is actually due to groundbreaking work done by her. So corn has benefited from a lot of just natural variation being generated by natural processes, and then humans capitalized on that natural variation due to things like transposons duplicating themselves. Soybeans. A soybean is a plant that is not a grass. So corn is a grass species. Soybeans are a dicot species. They're a legume. Legumes, there's a whole chapter in your textbook about legumes. Let's see, it is, what chapter is it? It's the chapter right after grasses, not surprisingly. Chapter 13 is about legumes. Legumes have a number of different characteristics that are important that you should know about. One of the most important of these is that they have a symbiotic relationship with bacteria in their roots. So in the roots, they have these things called root nodules. They're little outgrowths of the root. And in these root nodules, they're just packed full of bacteria. And these bacteria are nitrogen-fixing bacteria, which means that they take nitrogen out of the atmosphere that is actually double bonded, or triple bonded rather. Nitrogen in its natural state is 2 atoms of nitrogen bonded together with 3 bonds. And because they're bonded together with 3 bonds, this bond is very difficult to break. It's a very strong bond. And as a result of this, you need special enzymes to take nitrogen and take it out of its atmospheric gaseous form and convert it into a form that's actually absorbable by an organism. And these nitrogen-fixing bacteria have those enzymes. And so these bacteria live in the soil, and in some cases, these bacteria have formed these symbiotic relationships with legumes. And legumes provide bacteria with energy, and the bacteria provides the legume with nutrients. And the nutrient that it provides it with is nitrogen. And nitrogen is a key component of amino acids. And we'll talk about this more in detail when we talk about biogeochemical cycling. And so one of the things about legumes is legumes are really high protein plants because they have bacteria that give them nitrogen, which is a basic building block of amino acids. So legumes are important for human nutrition because if you eat beans, it's a good source of amino acids, of nitrogen. So unfortunately, soybeans have not been studied nearly as well as corn and wheat have been. So this is a paper from, oh, I can't see it very well. A paper from not very long ago looking at the evolutionary history of soybeans, once again relying primarily on a genetic analysis. And there are basically three possible ways that soybeans could have formed. So the modern soybean is called glycine max. And it's an interesting name because, of course, glycine is an amino acid. But presumably this came from a wild progenitor of soybean called glycine soja. And so glycine soja has much more genetic diversity than glycine max does, but it could have evolved through a single origin where people noticed that glycine soja was useful, and then they started cultivating it. And over the course of cultivation, the genetic diversity of soybeans has gone down a lot. And there are actually two different varieties of soybeans, in East Asian varieties and then American varieties that are very recent in terms of their origin. There's also the possibility that there's also been a lot of back crossing between the modern soybean G-Max and the ancestral soybean glycine soja. And so it could be that soybeans evolved from a single cultivation event. It could be that they evolved from multiple cultivation events, then followed by all of this back crossing. But it could also be that there were multiple origins that were separated by quite a lot of time. And once again, we're just early enough in really looking into the evolutionary history of soybeans that we're having difficulty deciding which of these evolutionary scenarios is probably the correct one. But in all cases, in all of these models, what is known is that there has been a lot of back crossing between the modern soybean and the ancestral progenitor of soybean. And that's what these arrows indicate, is gene flow from one species to another. As people have probably tried to get characteristics of the wild progenitor into the modern version of soybeans, there's been a lot of this back crossing, breeding this species with the wild progenitor. So soybeans is not very well studied yet, and so our understanding of its evolutionary history is very very in flux right now. Wheat, on the other hand, is very well understood. And so wheat, which evolved and was found naturally in the Fertile Crescent region, it's well known what has happened with wheat. And so we have wild wheat that was growing out in nature, einkorn wheat, and people started probably beginning to cultivate this because it has large, obvious seed heads that could be ground into flour, etc. And this was Triticum monococcum. And there's also Triticum circe, which is another variety of wild wheat. And these two species of wheat freely interbreed, but when they interbreed, it takes two diploid organisms, and when they interbreed, they don't divide their genomes, they don't divide their chromosomes, and so when they interbreed, they produce a hybrid that is tetraploid. So remember back to genetics. If you guys were wondering why we spent all of that time talking about genetics, remember that normally we think about organisms, when they go and do meiosis, they produce gametes with a single chromosome of each type in their in their genome. And then when these things breed with one another, you get a diploid offspring from diploid parents. So we have diploid parents, we have haploid gametes, and these haploid gametes produce diploid offspring. But in a number of different groups of plants, this doesn't happen as readily. And oftentimes what will happen is the diploid parents will produce diploid offspring, or diploid gametes rather, and then they will produce tetraploid offspring. Tetra meaning 4. Well, what happens when we breed these different varieties of wheat, these early progenitors of wheat, is we have one variety of wheat, which is einkorn wheat, and we breed it with another wild species of wheat. And what we get out of that is diploid gametes, and we get tetraploid offspring as a result. And so this first hybridization event represents a doubling of the genome. So that's why this little X2 is here. We end up with individuals that now are tetraploid rather than diploid, which is what happened before. And then probably through purposeful cultivation and breeding back to wild varieties of wheat around 10,000 years ago. Oh, sorry. So about 10,000 years ago is when we got this diploid wheat. And about 8,000 years ago, people took the emmer wheat, which is this tetraploid wheat, bred it with Triticum tauschayi, and got a hexaploid wheat. And so if we take this and cross it with another variety of wheat, we end up with wheat that now have 6 different types of genomes in them. This is hexaploid. So if diploid is you have 1 of each type of chromosome, diploid is you have 2 types of each chromosome, tetraploid is that you have 4 types of each chromosome, and hexaploid is that you have 6 copies of each kind of chromosome. And so when you eat wheat products now, when you eat a loaf of bread, it comes from modern wheat, which is Triticum aestivum, and this is a hexaploid hybrid that has developed over the last 10,000 years. And because the development of wheat has involved this increase in ploidy, it's been actually very easy to trace what its evolutionary history has been, which is why we understand more about the evolutionary history of wheat than any of the other domestic plant crops that we have. So it's 10:02. Let's just stop there, and we'll begin class on Friday talking about Jared Diamond's chapter. So if you haven't read that chapter yet, please do that. And I will send you an email when I'm done with your exams. I'm still working on them. It's been kind of a hectic week, but my goal is to get to that and get finished with that sooner rather than later. I'll email you a general message saying that they're graded, and then what I'll do is I'll send you out individual emails, and each email will have your test attached to it in its graded form, so you'll know how that went. Any questions about anything?
Where are these slides on Moodle?
They're in the September 30 folder. So let me go to that.
I don't see.
You don't see them?
No.
Well, I put them up earlier today. September 30, 2020 materials. Oh, crud, what happened there? I thought I had downloaded them into there. I'll do that momentarily. Yeah, I would have. I don't know what happened there. I could have sworn I did this morning. Anyway, I'll get them up, and then you'll have them. All right. In that case, if there are no other questions, I will see you on Friday.
I don't have a question, but
Yeah.
I accidentally turned my food blogging 2 into 3.
Okay.
So
or your food journal.
yeah. So
Okay,
I just put it into 2 today.
Okay. All right. Then I'll delete the one that's in 3.
Okay.
Okay. Cool.
All right.
Good enough.
Thank you.